selectable markers, and third one is first origin of replication, selectable markers. Third one is cloning site, cloning sites, and transfer the gene into the plant and animals. Transfer the gene into plants and animals. Okay. What is the character of plasmid? Plasmid is a single chromosomal double stranded, D for double, S for standard, circular, present in the cytoplasm of bacterial cell. Cytoplasm of bacterial cell is called what? Plasmid. Now this plasmid is used as a what? A vector. Plasmid is used as a vector. What is the meaning of vector? Vector is nothing but carry the gene from one cell to another cell. Carry the gene from one cell to another cell is called what? Vector. I should already discuss this one. Now, features of plasmid. These are circular. These are one strand, and then these are the next strand. It's our double stranded. Okay, and here I have what? Uri region. Then I have what? A Uri region. Okay, then in this side have a drop region. Okay, and this side have what? A antibiotic resistant genes, means ampicillin resistant genes. These are code to the what? PST1 and PVU1. Okay, and opposite to the ampicillin resistance, A for ampicillin, AMP means nothing but ampicillin. Ampicillin. Tetra. Opposite to the ampicillin have what? A tetracycline. TETR. In what is the tetra? Short form is TETR. In full form is what? A tetracycline. Tetracycline. R for resistance. Okay. Now these are code to the BAM H1 and SAL1. These are code to the BAM H1 and SAL1. Then next is in between the ampicillin and tetracycline have what? A EcoR region. Echerichia coli restriction enzyme 1. Okay, and then it's code to the what? A CLA1 and HIND3. CLA1 and HIND3. Okay, now ROP region is code to the PVU2. ROP region is code to the what? PVU2. And these are which plasmid? Plasmid, B for Bolivar or for Roderick, and the plasmid number is 322. These are about a plasmid. So outside these regions, all regions are called what? Restriction sites. Okay, and this region code to the what? Ampicillin resistant gene, and this region code to the what? Tetracycline resistance region. Okay. Now first one, origin of replication. What is the origin of replication now? After restriction enzyme cuts the DNA now. After cutting the DNA, we get the what? Number of fragments. That fragments are joined to the what? Uri region. Alien DNA. Point 1, origin of replication. Origin of replication means in which region have it replicated. In which region have it replicated? It's called what? Origin of replication. So which region? This region have what? Replicated. That's why alien DNA or foreign DNA ligate or joined to the what? Uri region. So alien DNA ligate means nothing but joint. Ligate to the ligate to the what? Ori region. Okay, because of this region only have a replication capacity. Then next one, selectable markers. Selectable markers. Selectable markers. Totally, how many selectable markers are there? 4 selectable markers. Ampicillin resistance and tetracycline resistance, and then chloramphenicol, and then kanamycin, These 4 are antibiotic genes. These 4 antibiotic genes are select the what? Transformed cell. It select the transformed cell. Select the transformed cell and eliminate the what? Untransformed cell. Eliminate the untransformed cell. Okay. So what is the transformed? Transform means nothing but these are the cell. Okay, these are what? A DNA. Okay, a piece of DNA introduced into the what? A host cell. A piece of DNA introduced into the what? A host cell is called what? Transformed cell. It's called what? A transformed cell. If this piece of DNA not introduced into the host cell, that is called what? Untransformed cell. Means simply absence of alien DNA. Absence of alien DNA in the host cell. In the host cell is called what? Untransformed. So the selectable marker select the what? Transform cell and eliminate the what? Untransformed cell. Okay. So these are what? Selectable markers. The next one is what? Cloning sites. Cloning sites. Okay. So how that transform cell is clone? So one cloning site is preferable. More than one cloning site means how many cloning site is preferable? Only one cloning site is preferable compared to what? More than one cloning sites. Okay. So in cloning sites, one cloning site is preferable compared to more than one cloning site. Compared to more than one cloning site. More than one cloning sites. Okay. The next one is what? See, in the cloning site, we select the what? Not we select that antibiotic genes are select the what? Transform and untransform cell. Select the transform cell and eliminate the untransform cell. In here, one procedure is there. That procedure is called what? A Combuser procedure. What it's called? Combuser procedure. Okay. So in this procedure, have a 2 plates. In this procedure, have a 2 plates. In one plate, we spread the water, ampicillin resistant gene. In one plate, we spread the water, ampicillin resistant gene. And another plate, we spread the water, tetracycline resistant gene. In another plate, spread the water, tetracycline resistant gene. Tet-R. Okay. Now, if we ligate the alien DNA at the tetracycline region of BAM-H1, if alien DNA ligate at which region? BAM-H1 region of tetracycline, here ligated. Okay. So these are what? Alien DNA. This blue color is what? Alien DNA. At which region we are ligated? BAM-H1 region of tetracycline. So what happened? In the presence of this alien DNA, this tetracycline is inactive. That's why this region is called what? Inactive insertion. Insertion, inactivation. We insert the alien DNA at which region? Tetracycline region. In the presence of alien DNA, this tetracycline is what? Inactive. That's why it's called what? Insertion, inactivation. Okay, next point is what? Next point is alien DNA. If alien DNA ligate at the alien DNA ligate If alien DNA ligate at the BAM H1 region of BAM H1 region of tetracycline resistant gene. So what happened? The tetracycline gene is inactive. The tetracycline gene is what? Inactive. Tetracycline gene. Tetracycline gene is inactive. Inactive. So it's called what? It's called what? Insertion inactivation. Insertion inactivation. It's called what? Insertion inactivation. Okay. Now this plasmid grow on the ampicillin resistant gene. What happened? And grow on the very tetracycline gene. What happened? Okay. Now if this plasmid grow on the where on the ampicillin resistant gene, what happened? Here ampicillin resistant gene is active because of not ligated at the what alien DNA at the ampicillin region. We are ligated at the very tetracycline region. So tetracycline is inactive but not what ampicillin resistant gene. Okay. So means this plasmid can grow on the ampicillin resistant gene. It can grow or cannot grow. If we culture on the ampicillin resistant plate, it can grow because of its resistance to the water ampicillin resistant. This ampicillin resistant gene is spread here now. Spread on the plate now. That's why already these are active. In here, this AMPR is active. That's why this if this plasmid culture on the where on the ampicillin resistant plate, it can grow because of what? This ampicillin resistant gene is what? Active. In active condition, it's have what? Resistance. That's why it can grow in the where in the ampicillin resistant plate. Okay. Then second, in second step, what happened? If this plasmid culture on the where on the tetracycline resistant plate, what happened? It lost the water resistance power. Na? Tetracycline is lost the resistance power in the presence of alien DNA. That's why no resistance power is there. Okay. Tetracycline lost the resistance power. So that's why no resistance power is there to the plasmid. Tetracycline resistant gene power is not there. Okay. Now, if this plasmid is culture on this plate, what happened? This plasmid cannot culture, cannot grow because of here lost the water this gene resistance. That's why in this plate cannot grow. Okay. So this composure procedure is a complicate. This composure procedure complicate. That's why alternative procedure is there. That alternative gene is there. That gene name is what? The gene name is beta galactosidase enzyme. That gene name is what? Beta galactosidase enzyme. Okay. So these are what are complicated because of simultaneously we want the what two plates, and on the plate spread the water tetracycline resistant gene and ampicillin resistant gene. Okay. Spontaneously spread the what the gene and spontaneously we want the water two plates. That's why these are what complicated. So alternative procedure is which one? Alternative procedure is which one? Use the water beta galactosidase enzyme. Use the water beta galactosidase enzyme. Okay. Now we see the water this beta galactosidase enzyme and their function and their nature. Okay. Beta galactosidase enzyme is a chromogenic in nature. Beta galactosidase enzyme is a chromogenic in nature. Okay. Chromogenic means nothing but gives a color. Chromogenic is nothing but gives a color. Okay. Alternative. Okay, gene. Alternative gene is which one? Okay, select alternative gene. Their function is what? Select the recombinant. Select the what? Alternative gene function is select the rDNA, recombinant DNA. Okay. Now this beta galactosidase. Okay, alternative gene is which one? Beta galactosidase. Beta galactosidase enzyme. Okay. Now this beta galactosidase enzyme, beta galactosidase enzyme is a chromogenic in nature. These are chromogenic in nature. Mean in the active condition, in the active condition, beta galactoside gives the which color? In the active condition, beta galactosidase gives the blue color, okay. Then in the absence of, not absence, in the inactive condition, in the inactive condition, beta galactosidase enzyme is a colorless, okay. Now if beta galactosidase insert, if the beta galactosidase is insert at the where? At the tetracycline region here, okay. So in the presence of this beta galactosidase, these are also inactive. Inactive. So in inactive condition, that beta galactosidase enzyme is what? Colorless. Beta galactosidase is what? Colorless. If this beta galactosidase, if alien DNA is not there, this beta galactosidase is what? Active. It gives what? Blue color. Okay. These are what? These are cloning sites. The next one is what? The gene is transferred into the where? Plants and animals. Okay. The gene is transferred into the where? Means which gene? Vector. That vector is there now. Plasmid. That plasmid is transferred into the water, plant and animals. Okay. Vector cloning into the where? Next step of vector cloning into the plant and animals. Vector cloning into plant and animals. Okay. Directly we discuss this. Agrobacterium tumefaciens. Cause the disease to this some dicot plants. It's caused the crown gall disease. Crown gall disease. Which one? Agrobacterium tumefaciens. Okay. Agrobacterium tumefaciens. Cause the disease to what? Disease to several dicot plants. Several dicot plants. So which disease caused crown gall disease? Okay. Now what happened? These are Agrobacterium. Okay. In the Agrobacterium have what? A plasmid. This Agrobacterium have what? A plasmid. In this plasmid have what? A gene is there. In the plasmid, this plasmid is called what? TI plus means in the gene, in the plasmid have what? A gene. The specific gene is there in the plasmid. Okay, this gene is called what? A TI. TI means it induces the water. Tumor. That's why what it's called tumor-inducing. Okay, tumor-inducing plasmid. Okay. Main in the Agrobacterium have what? Plasmid. That plasmid is called T plasmid generally. And in the plasmid have what? One specific gene is there. That gene is what? cause the what the inducing the tumor. That's why what is called tumor inducing plasmid. Okay. Now that means in the bacteria at the plasmid remove the water that gene which gene that Ti plasmid gene is removed from the plasmid. Ti gene is removed. Ti gene is removed from the plasmid and replaced by the normal gene and replaced by the what normal gene. Okay. Now here which gene is induce the tumor? That gene is what removed and replaced by the normal gene. Okay. Then that plasmid is introduced into the water host cell, nothing but plant. Okay. So it caused the tumor? No, because of we removed the water. That gene is removed. Ti gene is what removed. That's why no cause the what tumor into that plant when we are introduced the what that plasmid in the host cell. Okay. These are the 1 point. Okay. Right. The next one is what under this point mean here Agrobacterium. Again, I explain this one. Agrobacterium cause the disease. Agrobacterium tumefaciens cause the disease. The disease name is what Crohn gall disease. Okay. And in the bacteria have what plasmid. That plasmid have specific gene. That gene name is what a Ti means it induce the tumor. That's why that gene is called what Ti. T for tumor, I for inducing. Okay, when we cut the what the Ti gene from the plasmid and replaced by the normal gene and introduced into the what host plant. Okay, that is not cause the tumor because of that chemical releasing. Ti is release the chemicals. That chemicals are responsible to the tumor. So we remove the water that Ti sugar है गर्मी बहुत है ले लो कुछ ना बताओ okay so that chemicals released by the water the Ti gene okay that gene is removed from the what this plasmid. That's why that chemicals are not released in the host plant and not from the what the tumor. Okay, these are about in plants. Okay, then next is what in animals. Same. In plants, Agrobacterium tumefaciens cause the disease. In animals, retrovirus cause the disease. Retrovirus cause the disease, which is caused a cancer disease. Okay. Retrovirus cause the what? Cause the cancer. Cause the what? Cancer. Okay, same procedure. Okay. That cancer caused gene is removed. That cancer caused gene is removed and introduced into the what animal cell, nothing but human cell. Okay. Now that gene is what disarmed gene. Disarmed gene. Now that not with that gene is not cause the cancer because of we removed. Okay. These are what in the animals. Now next is what that recombinant DNA transfer in the host cell by the methods. Some methods are there. Yes, sir. Blur total. Okay. Next tool is what? Next tool. Make a competent to the host cell. Make a competent to host cell. Host cell. Make a competent to the host cell. These are the next tool. See? DNA, nothing but DNA. This DNA is a hydrophilic in nature. DNA is a hydrophilic. DNA is a hydrophilic in nature. Okay, that's why cannot pass easily into the host cell. DNA is a hydrophilic in nature. That's why what happened? Cannot pass or cannot transport, cannot pass or transport through the host cell. Cannot pass through the what? Host cell. So our aim is what? The DNA introduced into the host cell, but that host cell, our aim is DNA is introduced into the host cell, but this host cell is not receive the what? The DNA. So how this DNA is transferred into the host cell? Okay. Now some methods are there. First one, heat shock method. First method is heat shock method. Heat shock method. Heat shock method. And second method is what? Micro injection or calcium divalent ion. First one is calcium divalent ion. And third one is what? Micro injection method. And fourth one is biolistic method. Biolistic or gene gun method. Gene gun method. Okay. Biolistic or gene gun method. First one, heat shock method. First one, DNA is placed in the where? In the ice. DNA is placed on where? First on ice. Okay, then treated with what? 42 degrees centigrade temperature. Okay, and then again placed on the ice. Again placed on the ice. Now these are introduced into the what? Host cell easily. Okay, now rDNA introduced into the what? Host cell. Okay, the next one, first क्या किया अपन? DNA लिए, then DNA और DNA किस पे रखे अपन? आइस पे रखे. Then उसके बाद उसको क्या किया अपन? 42 डिग्री सेंटीग्रेड टेम्परेचर से ट्रीट किया. ट्रीट करने के बाद उसको अपन आइस पे रख के कूल करे. Then introduce into the what? Host cell. These are what? Heat shock method. Heat apply कर रहे. That's why it's called what? Heat shock method. Then next one is what? Calcium divalent ion. Second one is calcium divalent ion. Okay. For example, these are the bacterial cell wall and these are the plasma membrane. So already this cell wall is permeable to the what? All molecules. Cell wall is permeable to the molecules. Okay. Means easily allow the water DNA. But this plasma membrane does not allow the DNA. That's why this plasma membrane treated with the calcium divalent ion. When treated with the calcium divalent ion, here calcium pores will be open. Bacteria, calcium pores will be open. Through the calcium pores, the DNA is enter in the host cell. When we treated with the calcium ion, what happened? In the bacteria, calcium pores will be open. Okay, through the calcium pores, rDNA is introduced into the host cell or nothing but bacteria. The next one is what? Micro injection. Take a needle. Okay, take a needle first. Okay. Okay, in the needle, we take the what? rDNA. In the needle, we take the what? rDNA. Take the rDNA. Okay, and that rDNA directly inject into the what? Into the animal cell of the nucleus. This technique only possible in the where? In the animal cell, not to the plant cell, because of plant cell have a reached cell wall. That's why these are not possible into the plant cell. Okay, micro injection method possible only in animal cell. Okay, what happened in the micro injection method? rDNA taken in the where? In the needle. and directly inject into the directly inject into the what? And directly inject into the nucleus of the animal cell. Inject into the nucleus of the animal cell. Nucleus of the animal cell. Okay, these are what? Micro injection method. Then biolistic method. In biolistic method, what happened? Then the DNA, nothing but rDNA, labeled with the gold or tungsten. Labeled with the, these are labeled with the what? Gold or tungsten. Gold or tungsten. Okay. When we label the water gold or tungsten to the rDNA, their velocity will be increased. Okay, and introduced into the water plant cell. These are possible in the plant cell. Okay, these are make a complete to the host cell. Next, steps involved in the rDNA. Very, very important for 8 marks. And these all are tools also we discussed. These are also very, very important. Out of these 2 questions, compulsory 1 question is appear in the your board exam. Okay, one is tools of biotechnology, second one is what? Steps involved in the biotechnology. Okay, so tools of biotechnology is completed. In tools of biotechnology, what we discussed? First one we discussed about restriction enzymes. So, and nothing but not restriction enzyme, not only restriction enzymes, we discussed about enzymes. In enzymes, we discuss the ligase enzyme, we discuss the DNA tag polymerase enzyme, and we discuss the ligase enzyme. Okay, we discuss about 3 types of enzyme. First one is restriction enzymes. In restriction enzymes, we discuss the restriction exonuclease, endonuclease. Then polymerase enzyme, tag DNA polymerase enzyme. Then DNA ligase enzyme. Okay, these are about enzymes. Then next, what we discussed, then gel electrophoresis we discussed. Then next is what? Amplification of desired DNA fragment by using of PCR. ये भी discuss किए. Then next क्या discuss किया अपन? Plasmid discuss किया. Then make a comparative host cell. These are what in the tools of biotechnology. Next one, steps involved in the biotechnology. Okay, steps involved in the biotechnology. Link लेके आता हूँ. Link Okay, now steps involved in the biotechnology. Steps involved in biotechnology. Steps involved in biotechnology. Okay, means nothing but rDNA technology. Okay, step 1, isolation of DNA. Isolation of DNA. Then step 2, fragmentation of DNA. Step 2 is fragmentation of DNA. Okay, third step, fragmentation of DNA. And third step is what? Isolation of desired DNA fragment. 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 Step 4, amplification of desired DNA by using of PCR. Amplification or multiplication. Amplification of desired DNA fragment. Amplification of desired DNA fragment. Amplification of desired DNA fragment by using of PCR. By using of PCR. And the next one is ligate the DNA into the vector. Ligation is nothing but joint. Ligation the what? Desired DNA. Ligation of desired DNA into the vector. Ligation of desired DNA into the vector. And then now ligation of desired DNA plus vector is called what? Recombinant DNA. Now recombinant DNA transferred to the host cell. Recombinant rDNA transformed into the what? Into host cell. rDNA transformed to the host cell. And seventh one, obtain the foreign gene product. Obtain the foreign gene product. Obtain the foreign gene product. And last step is downstream process. Last step is downstream process. Downstream process. Okay. Now first step is isolation of DNA. We want the what? Isolation of DNA. For example, we want the what? Plant cell DNA or animal cell DNA or bacterial DNA. Okay, plant cell, animal cell or bacterial DNA or fungi DNA we want. Fungi DNA. Okay. For example, these are what? Plant cell wall. These are what? Cell wall. Below the cell wall have what? Plasma membrane. In the plasma membrane, inside the plasma membrane have what? Cytoplasm. In the cytoplasm have what? Nucleus. In the nucleus have what? DNA. Okay. In animal cell also. In animal cell outer layer is plasma membrane. Okay. Inside the plasma membrane have what? Cytoplasm. In the cytoplasm have cell organelle and macro and micromolecules. And also nucleus is there. In the nucleus have what? DNA. Okay. Then bacterial cell wall, bacterial plasma membrane. Okay, then cytoplasm, then DNA. Okay, bacterial DNA not bounded by the nuclear membrane. That's why it's a naked present in the cytoplasm. Then fungi. These are fungi, filamentous fungi. Okay, filamentous fungi. Inside the fungi have what? A cytoplasm, and then their nucleus, and then nuclear membrane, and then their DNA. We want the what? This DNA is. We want the what? This DNA is in the plant cell, animal cell, or bacterial fungi. For example, if you want the what? Plant cell. What is the outer layer of the plant cell? Cell wall, right? This cell wall is digested by the cellulase enzyme. Okay. Point 1 in the plant cell. In plant cell, cell wall. In plant cell wall is digested. Outer layer is cell wall now. So first cell wall is digested. Cell wall is digested by cellulase enzyme. Cell wall is digested by cellulase enzyme. Okay. Now below the cell wall, next is plasma membrane. Then this plasma membrane is digested by the detergent using of high detergents. Okay. plasma membrane, plasma membrane, plasma membrane is digested by what? Detergents. Plasma membrane is digested by the water detergents. Then in the cytoplasm, what macromolecules and micromolecules. Macromolecules are nothing but carbohydrates, okay, like proteins, okay, like lipids are present in the cytoplasm. Okay, these macromolecules are digested by their suitable enzymes. Means like a protein. In the protein, what is the suitable enzyme? Protease enzyme. Like lipids, what is the suitable enzyme to the lipids? Lipase enzyme. Then carbohydrates. What is the suitable enzyme to the carbohydrates? Carbohydratase. So these macromolecules are digested by their suitable enzymes. Okay. Then macromolecules are digested. These macromolecules are present within the cytoplasm. Okay, these macromolecules are digested. Macromolecules are digested by their suitable enzyme. Macromolecules are digested by their suitable enzymes. Okay. Now macromolecules also not there, break down by their suitable enzymes. Then nuclear membrane. This generally membrane is made up of what? Proteins and lipids. So by the protease and lipase, okay, is digested by the water nuclear membrane also. Okay. By what nuclear membrane also digested by nuclear membrane is also digested. Nuclear membrane also digested by their suitable enzyme. Their suitable enzyme. Okay. So nuclear membrane also digested. Then we get the DNA. We cannot take DNA directly because DNA is precipitated by adding of chilled ethanol. Okay, after adding the chilled ethanol, DNA get precipitated. Nothing but curd in form. Okay, curd kaisa rehta? वो form में convert होता. When we add the water chilled ethanol. Then in precipitated form, when precipitated form is convert after adding of chilled ethanol. Okay, chilled ethanol add करने के बाद DNA precipitated form में convert हो जाता. Then the DNA get or obtain or take by the spoon. That process called what? Spooling. Okay. Then first, okay. Then addition of after nuclear membrane, add the chilled ethanol. Add the chilled ethanol to the DNA. Okay, to the DNA. Then DNA get precipitated. DNA gets precipitated. Okay. After precipitation, DNA taken by the spoon. This process or such process. This process is called spooling. It's called what? Spooling. Okay. These are what about how we can get the DNA from the plant cell. Then animal cell. All are similar. All are again similar. Okay. Except what? Digestion of outer layer is dissimilar. Remaining all are similar points. Okay. Animal cell outer layer is plasma membrane. You know already plasma membrane digested by the detergents. Okay, this plasma membrane digested by detergents. Then cytoplasm is there. In the cytoplasm has what? Macromolecules. That macromolecules are digested by their suitable enzymes. Okay. Now that macromolecules are digested. Then nuclear membrane. That nuclear membrane also digested by their suitable enzymes. So nuclear membrane is also digested. Then DNA. The DNA cannot get directly. So adding the water chilled ethanol to the DNA. Then DNA get precipitated. Okay. After precipitation, this DNA taken by the water spoon. This process called what? Spooling. Okay. These are what about animal. Then bacterial DNA. See, bacterial DNA. Outer layer is cell wall, right? The cell wall is made up of peptidoglycan or pseudomurin. We learn in first year, which chapter? Biological classification. Okay. This cell wall is digested by the lysozyme enzyme. Okay, bacterial cell wall, bacterial cell wall, bacterial cell wall digested by what? A lysozyme enzyme. Lysozyme enzyme. Okay. After digestion of cell wall, then plasma membrane is there. That plasma membrane is digested by the what? Detergents. Plasma membrane is digested by PM. Nothing but plasma membrane digested by what? Same point. Digested by plasma membrane is digested by what? Plasma membrane digested by detergents. Okay, after digestion of plasma membrane, then cytoplasm is there. In the cytoplasm, macromolecules are digested by their nuclear suitable enzymes, and then DNA is there. Again the DNA get precipitated by adding of chilled ethanol and take by the water spoon. That process called spooling, common point. Then if you want the fungi DNA, for example, if you want the water fungi DNA, fungi cell wall is made up of chitin. Fungi cell wall is made up of chitin. That's why this chitin is digested or break down by the chitinase enzyme. Okay. So with the help of chitinase enzyme, this fungi cell wall is digested. Then below the cell wall, plasma membrane is there. That plasma membrane digested by their the water detergents. Okay, that plasma membrane also digested. Then cytoplasm, you know already, cytoplasm macromolecules digested by suitable enzymes. Then nuclear membrane digested. Then DNA get precipitated by adding of chilled ethanol, and after precipitation, that DNA can take by the spoon is called what? Spooling process. Okay, these are what? Isolation of DNA. Okay, now DNA is isolated. Okay, now DNA is isolated. That process completed. Now step 1 is completed. Then second step is what? Fragmentation of DNA. Then second step is fragmentation of DNA. Already we learn in a restriction endonuclease enzyme, right? Okay, this DNA, okay, this DNA incubate with the word. This DNA incubate with restriction endonuclease enzyme. Okay, so what happened to the restriction endonuclease enzyme? Cut the DNA into the fragments at the restriction sites, nothing but palindromic sequence site. Okay, now for example, here restriction site is there, cut it. Okay, now here also cut, here also cut. Where the restriction sites are at that region, DNA is cut and form what? Fragments like this. These fragments are formed. Okay, that's why what it's called, fragmentation of DNA. So fragmentation of DNA means DNA incubate with the restriction endonuclease enzyme. Restriction endonuclease enzyme. Okay, and the restriction endonuclease enzyme cuts the what? Restriction endonuclease enzyme cuts the DNA into fragments. Cuts the DNA into the what? Fragments. Okay. Now, third step. These are the second step. Third step is what? Isolation of desired DNA fragment with the help of gel electrophoresis. With the help of gel electrophoresis, we isolate the what? Desired DNA fragment. Okay. Already we discussed this point. In this, have what? One agarose plate. In third step, I'm telling third step. In this step, have what? Agarose plate. In the agarose plate has what? Fluid or matrix. That matrix name is called agarose. Okay, and has what? Small wells are present on the plate. Okay, this plate is filled with the, not plate of wells. These wells are filled with the water DNA fragments. Okay, then apply to the what? Electricity. Okay, so here DNA is there. That's why these are negative in charge. Okay, when we apply the water, electricity to the water, this plate, these DNA fragments are moved to the water anode region based on their size. Okay, which are small in size, they are reached to the water anode region. Okay, now this DNA is stained by the what? Ethidium bromide, and then immediately expose the UV radiation. So after exposing the UV radiation, the DNA fragments are appear which in color? Orange color bands. Orange color band. Okay, now this orange color band is separate from the water separate from orange color band separate from agrosplate. It's called elution. Okay. So we get the what a desired DNA by this gel electrophoresis. Then next is what a amplification of desired DNA fragment. Okay. By using a PCR. Here 3 steps are there in the amplification of desired DNA fragment by using a PCR. Already we learned this. Okay, this DNA is first treated with the high temperature. Here ATGC, AT, then TACG, TA. So in between have what hydrogen bonds. Already we discussed this. Okay. When treated with the high temperature, it's called denaturation, lost their property. Okay, after then, two DNA strands will be separate. Okay, after breakdown the water, hydrogen bonds. After breaking the water, hydrogen bond, these two DNA will be separate. Okay, then add the water primer to the 3' end. Okay, then add the water primer to the 2 sets of primer to the 3' end. And this process called annealing. This process is called what? Annealing. Already we discussed these points. Okay. And then annealing provides what nitrogen bases and polymerase DNA, tag DNA, Thermus aquaticus bacteria gives the Thermus aquaticus bacteria. In that bacteria have what a tag DNA polymerase. The tag DNA polymerase extend the what data primer and reach to the water 5' region, and here 3' to 5' region it's reached. So at the 30 cycle, billions of copies of desired DNA fragments are formed. These are about the amplification of desired DNA fragment by using a PCR. Then ligation of desired DNA into the vector. Ligation of desired DNA into the vector. For example, these are bacterial cell. Okay. Now these are plasmid. These are their DNA, cell wall, and then plasma membrane. Same point. We want the word plasmid. Means we want this plasmid. Plasmid is a vector, na? We want this plasmid. Okay. First digest the cell wall, by using of lysozyme enzyme, and then digest the water plasma membrane by using of detergents. Okay. Now this plasmid is a unopened. It's a circular. This plasmid is opened by using of restriction endonuclease enzyme. So which enzyme is cut the DNA? Same enzyme is used for cutting of plasmid in here also. Okay. So means in the presence of restriction endonuclease enzyme, this DNA, this plasmid is opened, and that alien DNA ligated the where at the what? At the plasmid. Now these are alien DNA. Now these are what? Vector. Okay. Now it's called what? rDNA. Recombination DNA. R means nothing but recombination. Combination of what? Two genes. One is alien DNA. Second one is what? Plasmid. So it's called recombination DNA. Okay. Now this DNA transfer into this. Next step is what? See. Ligation means nothing but joint. क्या joint कर रहे हैं अपन? Desired DNA को. किसको? Vector को. These are vector. These are desired DNA fragment. Okay. Now these are what? Ligation of desired DNA fragment into the vector. Then that rDNA transfer into the host cell. Now this rDNA transfer into the what? Host cell. Okay. So already we discussed this. rDNA कैसा transfer करे अपन? Like calcium divalent method, heat shock method. Already we discussed this heat shock method, and then micro injection method. Okay. Micro injection method, and then biolistic method. Okay. Now this rDNA transfer into the what? Host cell. Okay. After transferring the what rDNA into the host cell, then obtain the foreign gene product. Up now these are what? Replicated. Now these are what? Replicated and form what? A product now. These are express the what? The gene. Express the what? This gene. Their character is expressed here and form what? Product. In the obtain the gene product. In this step, what happened to that alien DNA or foreign DNA express and form what? A product in the 7th step. Then next is 8th step is what happened to when we release the market. These are important for 2 marks also. When release in the market, when product is released in the market, when product is released in the market, what happened? That product must be separated from unwanted material. Okay, that product must be separate from unwanted material and must be what? Separate and purify and preserve. Okay, what? Before releasing the market, downstream process I'm telling. Before release in the market, what happened? The product is separated. Okay, and purified after separation, purified and preserved. This step is called what? A downstream process, important for 2 marks also. These are about the steps involved in the biotechnology. So totally 5 LAQs are completed. 5 LAQs and some SAQs also completed. LAQs, glycol is completed, Krebs cycle is completed, C3 cycle is completed. And today and yesterday, what I discussed are this one, steps involved in the biotechnology and tools of biotechnology also completed, right? And only remaining 2 LAQs are there, tissue culture and then botanist. Okay. And after this class, we discuss about the SAQs and VSAQs totally, right?